exams going on. So she'll not be able to attend. So definitely she needs the recording. Yes. No, this is, yeah, I don't need that. So Aditya, you're recording on cloud, right?
Yeah.
Yeah. But anyway, Aditya, make sure that yeah. So Aditya, after every lecture, you have to download somewhere and then
Yes, I will share it to her.
lecture one and PowerPoint.
Yeah.
Okay.
Okay. Good morning, everyone. Lecture number 2. Okay. So, have you guys got the PDF of Leninger? Unless if you have a hard copy. Okay,
Yes, sorry.
Yes, sir. Yes, sir.
And the slides of lecture 1.
Yes, sir. Yes, sir.
Yes, sir.
Only pictures part. Anything which I write on the board of the page. Taking notes. Okay. So today we continue with the lecture 2. But last class I asked you a couple of questions. Okay. I think one of the questions is for example, like virus. Right? So we are talking about COVID pandemic. Okay. COVID is caused by what? By what? Okay. Anyone?
Sir, can you repeat the question?
Okay. What causes COVID?
Oh, it's SARS-CoV-2 virus.
Okay, it's 91. Okay. What is that? SARS?
SARS-CoV-2.
SARS-CoV-2. It is SARS-CoV-2.
Virus, right? Okay. I'm sure you have been reading a lot about this virus, right? So, can anyone tell me what is the size of this virus? Roughly. Any idea? 0.17. Maybe like how many nanometers? Roughly about 170 nanometers. Okay. So typically, so these coronaviruses, they have size anywhere from 60 to 140 nanometers. Okay, so that's the size of this virus, right? Okay, and then the next question which we wanted to discuss is that is it living? Is it living or not? Can you consider it living or not? That's the question. Because last class we defined elaborately how do we say something is living or not.
It is pseudo-living organism. COVID-19.
Yeah, so outside the body, it is like non-living.
When it enters the host, it tries to replicate itself. So
to circulate its biological process, it needs a host to regulate them. Until and unless they cannot regulate their biological process, until they get the host.
Yeah, yes, couple of answers. One says that pseudo-living, one says that okay, unless it enters the body, you cannot really call it sleep. Okay? But we are just talking about virus. Only virus, let's say outside, not infected. Once it's infected, of course, it replicates. It uses hosts metabolism, hosts machinery. We know that. it replicates. But however, let's say if it is existing by itself, like a bacteria, let's say on its own, okay, so then technically it is not living, okay, because what are the definitions which we used for living species? It needs to
reproduce.
it needs to, reproduction is one thing. Constant,
Metabolism.
constantly using
Constant energy and
Constant energy uptake and their conversion to their mechanical to do their works.
exactly constant uptake of energy to maintain its higher order structure. Okay, so working actually, against entropy means that entropy doesn't increase but it decreases because it becomes odd, right? So, but if you see virus alone on outside, okay, it doesn't really take any energy to do any work. Okay, it just exists just like that. So that's why we say that, like we know that, right? In WhatsApp or in video, you are saying that. Okay, so if you leave this virus on, let's say, any of the surfaces, then there are predictions like how many hours it really survives. Okay, if it is on plastic,
sir,
how
sir, I have one question, sir.
long, let's say cardboard, how long, etc., right? And we know that if you leave it long enough, even at room temperature, then this virus actually will die. That's because of the entropy, right? Let's say if it's stuck to a surface, like a wood, a plastic, or anything. So then you have interaction forces between the surface of the virus and the surface of the virus and the surface on it. This is eliminated, right? So
Can you repeat this again? Actually, voice is
Sir, the voice is echoing. The voice is echoing.
Can you repeat the last?
Can you please repeat this? Can you please repeat this?
Okay. So now it should be fine. Okay. So what was I saying? So let's say if a virus, so is on a surface. Okay. Let's say it is not in a living host. So then it's stuck to a surface. Then you can really think of interactions between the surface of the virus and the surface. And eventually these interactions have become strong and strong. Basically, it will destroy, okay, its organization. So typically, it's like, let's say, imagine a sphere of about, let's say, 100 nanometer. Then slowly, because of the interactions, it will just flatten and all the envelope will come out and whatever is inside spills out. So then it doesn't have its any architecture. So then basically it destroys its pure based on entropy. And then even weak interactions, okay, between the surface. What is the surface consist of? Basically protein molecules. On the proteins basically absorb, then membrane also will spread, and whatever is inside, the content comes out. Then basically virus is not in a condition which can even infect, because virus can, now we know because of a lot of information, the spike protein on the virus basically binds specifically ACE2 receptor on your lung cells. That's the only mode of entry into your body. Okay, so as long as virus is just on surface, then it will not really survive. So that's why we are saying that. So any surface, at room temperature, you can leave it for a couple of hours or a few days, then it will die. If it is elevated temperature, of course, then it will be much faster. So basically, technically, it's not living. Technically, it's not living. Of course, once it enters the host, then because of the host machinery, now energy is actually pumped in. So then it uses that machinery, then it replicates. Okay, but it replicates in a way different from our cells replicate. Okay, so that's one question I asked, right? I think if I remember correctly, I asked another question based on silicon. Right? So carbon can
Yes, sir.
catenated chains, okay? Sometimes it uses oxygen, sometimes it uses nitrogen, okay? Etc. But the question is that silicon. Silicon also belongs to the same, what do you call it, group, right? Okay, carbon silicon, just below that. So the properties, okay, what do you call it? Horizontally is period and vertically is group. Okay, in the same group, right? So basically, carbon and silicon have similar property, concatenation. So silicon can bond with oxygen, okay? Then it can actually form chains like this, right? So in that sense, the properties of carbon and silicon similar, okay? So you don't have basically most of the other elements to this. Of course, only phosphorus and sulfur, allotropes are there, but typically they are not really known. Okay. So then, what might be the reason why living systems don't really use silicon the way we use carbon, okay, to make organic life? What might be the reason?
Sir, when during our respiratory system, when we produce the CO2 in nature, CO2 is a single molecule, it can get outside our body easily. But when in case of SiO2 is a large three-dimensional polymeric in nature, and it is a solid structure. The expression of SiO2 will not be so as easy as CO2. So the reproducibility of the system will be lost.
But it's not that we take carbon only through CO2. We take carbon through CO2 route.
No.
Let's say protein inside our body. Sorry.
No, I am saying that when we are releasing CO2 from our body, it is kind of one part is the gas. Yes.
You have to think, for example, like our body is made up of molecules. We will of course discuss that. Let's say protein molecules. Okay. Or any bases organic molecules. You have a lot of carbon, right?
So because of catenation, because of catenation it has a tendency to form variety of products. So that can also be those products which are used for an organic for a living cell can be means because of catenation, sir, I think.
But that's the argument, right? Silicon also uses that sand. Okay, so silicon really makes long chains of molecules. Yes, I always use.
So silicon has a vacant d orbital, so it may be quite prone to nucleophilic reactions, which can break down that chain.
One of the major components in our body is water, so water can easily attack the silicon to break down macromolecule.
We need macromolecular structure which is flexible, I think.
So then what will happen to all the sand in the desert if it rains or the sand near the ocean? Why do you think it is stable there?
Sir, silicon oxygen bond is stable, but silicon bond are very much weak rather than carbon bond strength. So it is quite a cause that variety of biomolecules that is made up of carbon that can be made up of.
Sir, here in case of silicon, the importance of pi bonding is very much weak than that of the carbon. Carbon can easily, if we see the structure of the nucleosides, then there are many carbon-carbon bonds. But if Si is a double bond occurs, that will be very much weak. So that structure will not be stable.
We have to make protein. You know, what is our carbon source? Where do we get our carbon from?
During the making of protein, we need amino acids. So polypeptide chains will be made and then protein.
Amino acids, right?
Yes, sir.
So what are the bonds in amino acids? Carbon is bonded to what atoms?
Nitrogen.
Carbon is bonded to
NH2CO minus.
So one bond is carbon. Any other bonds?
Another C.
Carbon,
CO minus.
carbon hydrogen and carbon
oxygen.
Carbon oxygen.
CC.
Carbon
hydrogen.
These are the kind of important bonds, right? For example, in proteins. Of course, you have carbon. Okay. So these are the bonds, right? Okay. So now it's how strong are these bonds? Only if you know the bond energy, then you can really tell how strong are these bonds, right? So roughly these bond energies are ranging. Okay. So take my word, 350 to 400. Okay. Okay. So more or less, actually, all these bonds in a very small bracket. Okay, not really, not too big. Okay. But now you have to compare these bonds with the silicon. Now, if you replace, let's say, carbon with silicon in these things, okay. So what will be the bond strength?
Bond will be weaker.
Bond strength or bond energies. Of, let's say, silicon oxide. Silicon. Basically, you can really think of even silicon. Okay, let's say it is silicon oxygen or silicon nitrogen, silicon carbon, these bonds. So, what do you think these bond energies with respect to this?
Less in energy.
Weaker.
Yes.
More energy.
Some of them have more energy and some of them.
Yes, sir.
Some bonds are more stronger
Some
than
bonds will
carbon.
be stronger, I think.
Okay, one at a time. Who started first? Suman or Shubra? Don't remember this.
Aishwarya, sir.
Okay, Aishwarya, how much is roughly, let's say, these silicon energies, silicon oxygen, etc.?
Sir, silicon oxygen bond will be stronger than CO bond.
Strong, okay, we put one tick mark, okay, very strong, 2 tick marks, okay.
And I think similarly, if silicon, the electronegativity difference of silicon and nitrogen is more than that of the CN carbon and nitrogen. So I think that will be also stronger than carbon and nitrogen. I think for that reason, I think.
Okay,
And
let's see process. Okay, a lot of other people said Arijit, Prateek. Quickly.
So, sir, the silicon has some vacant orbital. So the bond has some double bond character, and the bond will be much more stronger than the other, that is silicon nitrogen or silicon oxygen, than compared to carbon nitrogen. So the bond, like in macromolecules or in living systems, the bonds are twisting that cannot be possible for silicon, because the carbon-carbon bond can easily twist single bond, whereas carbon oxygen, because silicon nitrogen double bond, silicon oxygen double bond cannot be twisted.
Okay. So what is your take? The bond energy is higher or lower?
Higher, bond energy is higher for a silicon oxygen and silicon nitrogen. I don't know about silicon carbon.
Okay. Okay. Arijit?
Yeah, other two have lower energy compared to the carbon hydrogen, carbon.
Okay. Okay, fine. But let's not worry about okay, so we can always. Okay. So but one of the important bonds is basically oxygen, right? Okay. So in most of the molecules. Okay. So acids, right? Acids you have COO. Okay. So if you consider that, so these energies, okay, not too weak, but not as strong as these bonds. Okay. So somehow when the life evolved, or before even molecules are formed, all types of molecules are formed, strong, weak, etc. But when the life molecules, for example, one of the hypotheses is that RNA overload, okay, you can really reason on that. Okay, so some polymers, okay, based on DNA-like polymers, okay, most of the people think that initially some amino acids were formed, somehow they're put together, bases were formed, they're together, no. So in those molecules, okay, so these bonds are there because the energy is strong enough, okay, that they exist for some time. But at the same time, they are not too strong. If they are too strong, then you will not be able to break them. Okay, so they are so rigid Okay, so then you lose the flexibility to mold them into the molecules which you want to be. For example, this is so strong that the moment you have silicon oxygen, okay, so this is roughly twice as strong as this. Okay, so approximately 800. So now you can really think of the moment you have to use oxygen as well with silicon. Now you have really trouble. Okay, you know the average temperature. Okay, for example, like we are adapted to physiological temperature, like inside our body is 37 degrees Celsius. But even if you take outside, okay, so roughly ballpark, okay, average temperature is let's say 22, let's say 30 degrees Celsius. So then you can really think of these bonds versus these bonds. Okay. So though energy is only twice, right? But we know that from the kinetic point of view, twice the energy is really bad enough that we can't really do. But at the same time, these bonds, okay, so more or less all are tightly packed in very small bag region. Okay. So that somehow made that carbon, in addition to, of course, it has the property of concatenation. And also it can really combine with these basic elements, which really exist in ample amounts, okay, oxygen, hydrogen, nitrogen. So made somehow life evolved with, okay? But it's possible that elsewhere maybe we can really think of silicon-based life. Okay, that's one of the arguments, is basically based on the energy. So these bonds are so high that somehow our conditions are not appropriate, okay, to use this, because you need flexibility. So that sometimes you have to break down the big molecules into small molecules. That's what happens. Sometimes unused proteins, okay, misfolded proteins, need to be broken down to individual. Then you need to really break carbon-nitrogen bond, carbon-carbon bond, carbon-oxygen bond, or you do some chemistry, you know. So then they should be accessible. But with silicon oxygen, definitely it's not really possible because it's much higher than that. Somehow, so life will want that. That's roughly kind of answer to question number 2. This was eluded, but some other textbooks have actually allowed it. So depending on which textbook you use. But online, you can maybe access this.
Sir.
Yes.
So, sir, I say that the carbon has only one isotope, whereas silicon has many more isotopes. That's the reason that the nature choose carbon, because if many more isotope of silicon compound come into the body, then it will a lot of trouble in the systems, whereas carbon only is mainly 99% is 12 other isotopes.
So for example, let's say in the nature, if C13 is available, okay, in plenty, you can still have life. It's not going to affect you. It's only because its abundance is low, only 1% C13 is there. So in your body also you have basically, okay, only 1% C13, okay, because you use natural resources, right? But isotopes, the reactivity is not going to be used, okay? So it's so it doesn't really affect much. Okay, how do we know that? So we make actually proteins, okay, in test tubes based on completely enriched isotopes like C13, N15, okay, and even for DNA. Okay, based on those information. So isotopes don't really change properties a lot. Okay, the reactivity a lot. Okay. For example, NMR is the best example, nuclear magnetic resonance. You solve the structure of protein molecules. So you use actually isotope labeling. Okay. Really use all carbons, C13, all nitrogens, N15, you can make a molecule, and the properties of the molecules are same as your natural abundance carbon and nitrogen. So based on that argument, I can really say that the isotope abundance is not going to be a big factor. It's not going to change much. Okay. So okay, come back to. Yes?
So I have one question. So if in case we want a silicon life on Earth, what should be the medium and temperature of the Earth which makes the silicon life appropriate on Earth? Means the conditions which will favor the formation of silicon-based life.
Exactly. So that's the appropriate question. So for example, like the conditions on Earth, okay, prebiotic conditions, okay. So water, properties of water, the temperature, and any other perturbations like electric discharge, okay, lighting, etc. So started making all these molecules. So you have plenty of molecules, okay, formed based on carbon, because it's easy to break down, make thing. At the moment with silicon, so current conditions on Earth doesn't really allow you to make silicon-based molecules plenty complex. Okay, can you really name molecules based on silicon versus molecule based on carbon? Molecule based on carbon, you can really think of any organic matter. Okay, so you can mix a couple of carbons, nitrogens, oxygens, plenty. But you can't really do that silicon. So you don't have that heterogeneous mixture of silicon-based compound. So then the question doesn't really arise that, so unless you have those molecules, so somehow now you put it into that, say, okay, catalysis cooker to make these molecules. Like you mentioned that it's possible that if you have, let's say, very much higher temperature, imagine much higher average temperature, okay, or some other planet. So then maybe now you can really begin to think of breaking SiO bond at will, but then of course these molecules will be not stable. Okay, then you can really think that these are like, now we say covalent bonds, stronger bonds, and quote unquote they'll become like weak van der Waals. Okay, so think of that. That's a possibility. So that's what actually we look outside of planetary system or solar system for these kind of molecules, where maybe silicon-based life is possible, but not the current conditions. It could be maybe higher temperature, maybe higher pressure, okay, and maybe even different solvent. What is somehow appropriate for, because you need to solubilize, right? Because water is basically the solvent where the molecules move very easily, diffuse, collide, make new molecules, different geometries, different compositions. Once you have this lot of combinations, then by chance, maybe then you can really make more complex molecules, and then slowly they accumulate, quote unquote, what we call life. Okay. So yeah, let's come back to now today's lecture. So again, start with the size of cells. Okay. So cells. Okay. So we briefly touched upon basically with the picture which I showed on the slide, prokaryote and eukaryotes. Prokaryotes are basically, let's say, for example, bacterium has about a 2 micron long and about a little less than a micron diameter. Okay, and it has flagella and villi. But the mammalian cells are the multicellular organisms more complex because they not only have the individual cells, but they communicate with each other to actually enrich basically their life. So then how do you categorize cells? So if it is bacterium, very simple. Okay, you have one size, only one type. There are no different kinds of cells. Okay, of course, there are certain bacteria which have what's called social life, means that these bacteria actually work together. Okay, for reasons that somehow they can actually share the nutrients. Okay, or somehow if they pack tightly. Okay, these are individual cells, but still because of the social behavior, imagine like when you have a party, many people come together and share food and etc. So bacteria also actually certain bacteria. Okay, so you want to know the name of the bacteria? Read the first chapter of literature, then you will find it. If there is a name there, you have to find it. Okay. But otherwise, so the prokaryotes or unicellular cells have independent, okay, and single type. But multicellular organisms like us, okay, so where you have different types. Okay. So now because there are different types of cells, then you have to begin classification or categorization. So then what are the properties by which you now separate or you can use size, shape, okay. So how big or small, what are these shape, and then lifespan. It's not that all cells actually in our body have the same lifetime. Okay, some cells short-lived, some cells have life as long as you live. Okay, so these are the couple of the properties. Okay, and so when it comes to size, okay, so roughly bacterium we know. Okay, it's about a micron, 2 micron long and above this, right? But mammalian cells are eukaryotic cells. Okay, so we are talking about now eukaryotic. Okay, so the name eukaryotic and prokaryotic means that karyotic means nucleus. Okay, so eukaryotic means that U means in Greek word true, means that really they have a true nucleus. Prokaryotic means before nucleus, means that they don't have a well-defined nucleus. Okay, so let's say if you come back to eukaryotic cells, like cells in us. Okay, what are the typical sizes? So roughly average size is about anywhere around 5 to I would say 100 microns or microns. Okay, that's the size. Okay, that's the average size. But it's not that all cells are exactly of the same size. Okay. So then you can really ask, what is the smallest cells? And what is the largest? Because it's always good to know that. Please. Okay. So, what do you think is the smallest cell in our body, and which cells are the largest?
So, nerve cells are the largest. Nerve cells are the largest cells. Nerve cells, the neurons.
Okay, Tushar, you are saying neurons are the largest.
Yes, the largest cells.
Okay, so we will come back what's called okay. Shortest or the longest, we come there. Okay. So neurons are the longest cells. Okay, they are really long. Okay. So, but we are talking about the size.
The skin cells. Skin cells are the largest.
Okay. Yeah.
Skin cells. Skin cells are the largest.
Yeah, those are the largest in the body. So roughly, what is the size?
Is the length of the eye cell?
Yes, Pratish, length of the eye cell.
Yeah, I mean, I may have heard about it. I'm not sure.
Okay.
Is it a cell or I don't know.
So smallest are one are called they are in our blood called platelets. You might have heard the word. Okay. So our blood, okay, is considered a tissue. Surprisingly, though it is liquid, it has different types of cells. One is obviously red blood cells, which we know, and white blood cells. So white blood cells, again, different category. There are a couple of categories: neutrophils, platelets, basophils, eosinophils, and whatnot. Okay, platelets are the cells, okay, which come into action when you have a small blood cut. Okay, whenever there is a blood leak, immediately the platelets will come to the place where the blood is leaking, and then they initiate the process of clotting, blood clotting. Okay, so the initiators are platelets. So these are kind of smaller in size, and then size is about 4 microns. Okay, that's basically kind of on the smaller size. It could be anywhere from 2 to 4. Okay, so again, these numbers are not very rigid. Means that you can't say that, okay, I found a cell which is 4.5 micron, but you said that it's 2 micron. But we are talking about range. Okay. the largest cell. Okay, and another category also in platelet cells is sperm cells. Okay, sperm cells also, which come under we call what the meats. Okay, so they are also smallest in the sense that you know how the sperm looks like, right? It has a body and it has a light tail. We are talking about the body. Body is 4 micron. Okay, but the tail is very long. Tail is roughly 10 times longer than body size. So you can really think of that's like 40 to 50 micron. So if you don't consider the tail, then the body part is. So these are the small. So most of our cells are really large. What are the largest cells in the body? Anyone? Okay. So the large means that I'm talking about the volume. So we'll come back to the longest. Longest are neurons. Neurons or neurons. Okay. The largest again gametes. But what is the name of that gamete?
Excel.
Ovum. Is it ovum?
Ovum.
Ovum.
Excel.
Ovum cell are basically egg cells. Okay.
Egg cells.
Okay. So roughly, what do you think size of the human female egg cells? We're saying these are really longest, the largest, right? So we have given roughly the number. Okay. So this is roughly about 200 microns. Means 0.2 millimeter. Okay. 0.2 millimeter means that now these cells can be really manipulated with tiniest tweezers, okay, in labs. Okay, so where you can really put a glass micropipette and then you can actually inject things into this. Okay, that's where how it's done in vitro fertilization. Okay, because these cells are really large enough that you can really manipulate this. Okay, so but in real life, have you seen even larger eggs?
Yeah.
Yes, Mr.
In terms of large cells, what about zygote?
Zygote. So zygote you call zygote is how do you define zygote after fertilization?
Which is the first fertilization?
Yes. So that's basically you have to combine one ovum and one sperm.
Yeah.
So sperm is very tiny. So ovum is more or less. So if you combine, basically set. But otherwise, individual cells we are talking about. So that's of course where the life begins. You can really consider that as the zygote. But zygote size is this. Everyone is seeing my slide share. Sorry, you didn't get a laptop. Maybe you can really see that.
Yes, sir.
Okay, so you see the egg, right? This is really the chicken egg. Chewy. So what is the size of this? This is a single cell. Okay. So roughly what is the dimensions? Longest thing. About 10 centimeters. 5 to 10, something like that. Okay. So this is a single cell, by the way. Okay. This is
Sir, why do we call this chicken egg a single cell? Because whatever we usually know about cell, there is a nucleus, cytoplasm, and things like that.
exactly. So here also, so what you don't really see is that so okay, every cell has how many sets of chromosomes? Only one cell, right? Every cell has one.
Okay.
Chicken egg also has one. Okay, so but that's so tiny compared to your yolk. Okay, so if you go to actually Google and then just type egg and nucleus, you can really see that some pictures really show you the tiny white spot on this yolk. Okay, but you have a lot of fat that's yellow. Okay, along with some protein, and then you have albumin, all the protein content also. Okay, but this is typically an egg cell. Okay, so so that's the size. Okay, smallest, largest, longest are neurons. Okay, so the longest neuron is in your body. It's roughly about a meter. Okay, so it goes from the base of your spinal cord, okay, back base to the big toe. That's about a meter long. Okay, imagine that's a single cell. Okay, so single cell, it has a huge cable, okay, called axon. So neuron cells basically have, we'll talk about the rough content, but it has this, but then it has a long cable. Okay, so where it communicates. Okay, so basically that's your nucleus and the cell body. But then all everything is single cell, so this is called axon. Okay, so that's where basically the nerve signal transmits from the cell body to the synapse, and this is where actually communicates with another cell. Okay, and then there is the transmission of signal because of whatever actually it releases. It releases sometimes calcium, sometimes some other small molecules, etc. Messengers, secondary messages. So neurons are the longest. Shortest means that smallest, right? The sperm cells. Okay. So this brings me the shape. Okay, the shape of neurons are like this. So you have cell body, and then you have a long cable called axon. And so what are other shapes we have? Okay, so other shapes, for example, okay, so shape. Very long neurons. Okay, so but if you take red blood cells, RBCs. Okay, so they have very interesting biconcave shape. They are kind of disc shape, means that so they have a concave, okay, shape and a disc shape. So this is basically if you cut in half, this is what actually it will look like. So they have a flattened. So you have basically the center of the cell, a depression. Okay, so that's very called concave. And these are again roughly of the size of 8 micron in length, diameter, and then thickness is about 2 micron. Okay, so the shape of RBCs is actually very interesting. Somehow this shape actually helps RBCs. Because of this biconcave shape, they can actually what is called bend. They can actually bend very easily, and then just like a roti, thick paratha, stuffed paratha. you can actually really bend very easily. Why that is important? Because the red blood cells are in the circulatory system in the blood. Imagine when they go to capillaries, okay? They have to go in tissues, the capillaries become narrower and narrower, okay? So when they go to narrower spaces, the diameter of the capillary actually decreases. Still, the RBCs actually can go through these small, narrow spaces because of the buckle. Imagine if it is a very rigid disc, then you have to have a capillary at least larger than this, okay? And imagine if one RBC gets stuck, then nothing can, it's like a clogging a pipe, nothing can do it. But because of the buckling shape, they can actually navigate this. And so roughly the other cells in the red blood cells, like platelets, they're actually very tiny, okay? And of course, the number of RBCs is much larger. Okay, so that's
in the red blood cell, if there's something to maintain the shape, because we know that if we have a certain body,
yeah,
it will always take the shape of the smallest surface area. In such cases, the speed.
Smallest surface area. Okay.
Correct. It takes shape of the smallest. It takes the shape of the smallest surface.
Yeah. So how actually shape is maintained? So there is a protein called. So there is a huge cytoskeletal network. Okay, network formed by a protein called spectrin. Okay, along with other intermediate filaments. This actually makes the shape. Okay, so at the same time it's fluid. Unlike other cells, where actually cell shape is not modulated very easily. Okay. So what are the other cells? Okay, so this is one type of shape, and this is one shape. And then if you take the muscle cells, okay, so which are in your three types of muscles. One is heart muscle called cardiac muscle, and another is called connective tissue. Okay, so where actually they maintain elements between two cells, are active muscle. Okay, so any muscle which you use to actually bang your hand, okay, or when you speak, your vocal cords, or actually when you digest your food, okay, so you have to actually move your tissue. So all these cells come under different types of muscle. So this muscular tissue, okay, because they need to really exert force actively, okay, so they are under constant tension. Okay, heart of course 24 hours is working, okay, breathing, so that tissue is active all the time. But some tissues in your body, they are active. For example, when you run, certain types of tissues are active, or when you digest food, certain tissues are active. So these cells, muscular cells, are typically very long, okay, and these are like inside there are, okay, so filaments, and this actually now can compress. Okay, so it's called contraction. Contract elements, contraction, okay, relaxation, relax. So the entire cell actually becomes elongated. You can really call elastic. Okay, so these cells constantly, they're compressed, they're stretched. That's what really happens when you do. Okay, pump your pump iron, you get muscle. So they have slightly different shape. Okay, so different cells. Now see how much rich we got into when we moved away from single cell to multicellular system. So each cell in different tissue has its size and shape. Okay, so now comes to the lifespan. Okay, so what are the shortest living cells? And longest living cells. So which cells do you think that in your body are have very short life, and which cells have really long life?
RBCs are the shortest one, shortest living cell.
Okay, RBCs. What is the lifetime?
3 days is 3 days.
That's
No,
too
it's
short.
I'm not sure
WBC.
about
2 days is too short. Okay. This is really.
W
120
WBC.
days.
Yeah, this is 120.
WBCs. So WBCs have specifically 1 week of lifespan. WBCs.
White blood cells.
Yeah. Those have specifically 5 to 7 days their lifespan.
So RBC is slightly longer. Okay, so they are roughly average is 120 days. Okay, average is 120 days. Anywhere from you can really say 60 to 140 days. Okay,
Isn't it
so
a stem
there is something called neutrophils. Neutrophils, part of white blood cells, they live on an average 2 days. Every 2 days, you replace your neutrophils. Okay? Means that they don't retain memory. Okay? So, but some other white blood cells have to have a long life because there is some memory. Okay? So, these are the shortest you can really think of. Okay? But not all cells, I mean, imagine if everything needs to be replaced 2 days, then you have to leave. Okay, so what are the longest living cells in your body? Which cells do you think that you say that, oh,
Heart muscle cell.
good and still I have this?
Heart muscle cell.
Heart muscle cells. Unfortunately, heart muscle cells actually kind of not so short, but replaced continuously.
Neurons.
The neurons are actually very long lived.
Neurons are one of the long lived, but not all neurons.
Sperms.
Neurons, okay, they fit, but even there are long lived cells.
Versus sperms can also live long.
Who said that? Abhijit?
Me, Pradeep.
Pradeep, yeah. What cells?
Sperm cells.
Sperm cells.
There is the DNA containing that part. There is the DNA to live a longer time. That the DNA containing this part.
No, but we are talking about whole cell. Imagine, let's say, we are talking about individual cells. Let's say individual sperm cells. How long do you think that they live? Again, they don't have really long life. Definitely not this.
Cells of ice. Cells of ice. There are some cells in ice, so they never decay.
So island cells. So we have lens. Okay. So it's called lens because it literally looks like a glass. Okay, transparent glass. But it's made up of cells, and these are rich in protein called crystalline protein. Okay, highland cells. These are rich in crystalline. As the name suggests, these proteins are so dense. Okay, inside these cells, it's like crystals. Okay, so very little, aqueous content is there. Because they pack so nicely, they look transparent. Okay, and any crystals which you see like sugar crystal, okay, salt crystal, when it's perfect It's like a transparent cube, right? Proteins are also exactly. So island cells, more or less, they are actually all your entire life. Okay, the moment you are born, okay, more or less, the eyeball is actually formed. Very little change really occurs throughout your life. Whole body grows, but typically the eye is already, the size doesn't really change much. So the cells in islands are the longest really. Okay, but neurons are also correct. Okay, so I want to really show something. So where, so this one. I'll send this PPT. Okay. Show. Okay. Doing this one. So where is the time? What cells in the human body live the longest? Okay. So they have done these experiments. Okay. So like I said, neutrophils are the shortest lived. They have only 2 days. On an average, your body replaces roughly average is 7 to 10 years. But neutrophils are 2 days. And the maximum, they have done this experiment, so they have actually replaced brain cells, okay, from old mouse to another younger mouse, and then they really survived the life of the younger mouse entirely. So looks like, in principle, the brain cells can be, what is the word? Let's say move from one body to another body, they can be done. Or that's one example. Okay, but we are talking about within a living organism. But here actually they have shown that they really survive. Brain cells 200 years. Okay, islands lifetime, island cells, egg cells 50 years. Okay, so egg cells, not the sperm cells. Sperm cells are really short. Egg cells are long lived. Heart muscle cells, some muscle cells, okay, are 40. Intestinal cells, okay, excluding the lining. Lining is replaced continuously. But some intestine is like 16 years. Amazing. Skeletal muscle, some muscle cells, not all muscles. Because some muscle you have to build. If you don't exert pressure, actually your muscle goes away. There's something called muscle wasting. So if you have read stories about the astronauts in space, because it's zero gravity conditions, there's no gravity, so there's no, weight, okay, so it's weightlessness conditions. So you actually lose your muscle, because muscle importance is that you actually, because of the inertia, you move, you exert energy, but if you don't have to, you don't need muscle. So they lose muscle in space stations. So they have actually constantly to do this exercise, So that's why when actually after they spend a lot of time in space stations, when they come back to Earth, it's not easy for them to actually, they have to really build muscle back. So it's very, it's not easy, okay? So fat cells 8 years, hematopoietic cells 5 years, liver cells, okay, partly. Okay, so that's basically the lifespan. And roughly now we have seen the size of the cells. Right? So bacteria are the shortest. But anyway, so you can see now about 1 to let's say 100 microns. Okay, cell size. Why can't we have size less than a micron? Let's say 0.1 micron. Why not? Can we have cells smaller than this? Or not? Can we have cells smaller than a micron?
Sir, it might not have that much of cell organelle that that should be there.
Yeah, can you elaborate? That's the right approach. So what happens that if I try to reduce the cell size to less than a micron, I'm thinking even smaller than a bacteria. Why can't I? Yeah, you said that yes, cell organelles. Can you elaborate?
Yes, sir. That's what I was saying. That might be cell organelles might be absent, substant cell organelle, and there might be only genetic material and some sort of specific organelles present. I think.
Okay. Yeah. So we can't have very small cells because we need to have, we see the content, we need to have at least nucleus. We need to have other periphery machinery. Okay. So nucleus itself is typically a micron in size, eukaryotic cells. Because we have 46 chromosomes, we have to pack all those things. So the size itself is a micron. So you can't really have shorter. And the same thing for bacterium. So bacterium already packed, okay, without having any nuclear envelope or nuclear membrane. Everything is packed in tiny tube. So now you can't really reduce. If you reduce, then you won't have proteins. So if you won't have enough proteins, then it will not use. So there is a minimum size required. Okay, why can't we have larger cells?
Sir,
Let's say 1 millimeter. Or
sir,
okay, so short we understand because we can't pack everything into the tiny volume. So you need to have minimum size. Okay, that's kind of.
Sir, I have a question, sir.
Yes.
Sir, bacterium cell, they take the oxygen for respiration through diffusion.
Okay.
And so there should be a concentration gradient inside the cell and outside the cell of the oxygen. So if the outside of the cell, the concentration of O2 is less, then the cells are decreasing the concentration by increasing the volume. So can we say that they are changing the size according to the condition of surrounding?
No, I mean, see that there's no harm. Okay, so let's say if it is 1 micron. Okay, so we are talking about smaller size. Okay, so we know that now we cannot really put everything. Okay, so next question is that, yeah, larger size. Okay, why can't bacterium have a diameter just one micron, but it can be long, right? Then your accessibility of oxygen is solved. Long tube, like axon in nerve cell.
Okay, sir. Means they are elongating cylindrically, or they are not.
That's one way to solve your problem of getting enough oxygen. Okay. So, but let's say why can't we have mammalian cells? The largest basically egg cells, but typically let's say that's on higher. But why can't we have the 1 millimeter? Let's say 10 times larger, 1000 micron. What happens if we have a millimeter cell?
Sir, what I got it, they are elongating cylindrically. That's why the size of their cell will remain same. The diameter will remain same, and so that we can say that a cell have a particular diameter size in different conditions.
No.
Am I correct, sir?
We have different problems. See, one problem is that they need to get the nutrients. Okay, and part of the nutrient is oxygen and nutrients. Okay, anything else which they need to take out from the environment, they have to take it. But the thing is that of course they have to move as well. Right? So of course, if bacteria becomes really long, then of course the mobility will be a problem. You know, right now it has the flagellum. Let's say you have a bacterium. Okay, so it has a flagellum which is actually optimized. Okay, so for this size, but of course, if you have, let's say, twice the size, because then you need to have your motor also needs to be strong, right? Of course, then you need to have maybe longer flagella express, but somehow they are optimized. Okay, so it's not only taking nutrients, but the mobility is equally important. Okay.
Okay.
So let's so bacteria somehow optimize. That's okay. But mammalians, the problem. What is the problem? Why can't we have millimeter cells?
So if the metabolic process needs the oxygen, so continuous diffusion of oxygen in the cell is required. So the rate of diffusion of oxygen will be much more slower, much more smaller if the cell size is so much bigger. In that case, so we can't have that much big.
One more.
Yeah. So one of the main concern is that. So concern is basically, yes, getting the nutrients into cells. Okay. So not only just oxygen, a lot of other things. So if cell is really, I'm just writing only cell. Okay. So imagine something needs to move from one end of the cell to the other end of the cell for some reason. Okay. Could be small molecule, or it could be large vesicle, etc. So within the cell, you need to really move here to here. Okay, right? So then you have to really find out how to do that. Okay, but you can always ask the question already. In neurons, we have this. The mater of neuron. Then imagine it's a single cell. So whatever happens at the somewhere in the nucleus, okay, or near the nucleus, need to be communicated. Okay, that we are doing to electrical signaling. Okay, that's a totally different path. But let's say if you want to, but you don't want to have that every cell. Every cell then is really long, then crisscross. Then you're actually it's not optimized. Okay, so only neurons. They need to be really long because they need to really communicate. The signaling needs to be very fast. Okay. Imagine something really just pinches in your big toe, suddenly you know, right? But if that's because of electrical signaling. But everything cannot be just electrical signaling because your environment, your input is food or molecules, visualization, photons. You can really think of sound, So there are many different 5 senses we have, right? So it's not only just the touch, but there are a lot of other senses, So you need to basically accommodate mechanisms to work around problems. And for almost everything, you need to really move material, not only from outside cell to inside the cell, but within the cell. Within the cell, whatever happens in the nucleus, we'll come back to that. Nucleus needs to be finally conveyed to the edge of the cell. Sometimes you have to move material from the edge of the cell to the other edge of the cell. Okay, so then basically your limitation is now this side. Okay, and so within the cell, of course, from the outside the cell, again you have a lot of inputs. Okay, so you can really say that your inputs can be, let's say, a molecule A, B, C, D. You have actually receptors sitting in the cell. Okay, and then you have transporters. Okay, good. So the thing is that you can't have transporters for every small thing. So something is very, very general. Then you want to use what's called diffusion. Okay, so partly you use diffusion, but partly you use active transport. Diffusion versus active transport. How do you distinguish? Diffusion means that you don't have any active mechanism, say that you have to really go from point A to point B. So if you leave it here, so basically they diffuse in all directions. Okay, so there is no specific direction. That's diffusion. But let's say it is active transport. You know that, okay, from A to B I need to go. Molecule A need to go from A to B. But molecule B need to go from, let's say, capital C to, let's say, capital B. So then you need to really have a mechanism that directional motion. Okay, so cell has actually this, but you need diffusion also. For oxygen, it relies completely on diffusion. Every cell relies oxygen on diffusion because you don't have any oxygen transporters. Okay, within the cell, say that okay, oxygen entered here. Okay, it needs to be taken because it needs to be available almost everywhere. If it is required everywhere, you don't need to transport, right? It should be available everywhere. If it is available everywhere, best is that leave it to diffusion. But now oxygen is available. Imagine your blood vessel. Okay, capillary is passing only here. Okay, so then your RBCs are actually going through this capillary. Okay, so now the blood came from heart. RBCs have a lot of oxygen. Now they leave oxygen here. Okay, they won't leave oxygen inside the cell. So they leave somewhere here, and then this oxygen needs to diffuse. Okay, so it's easy for it to diffuse some distance beyond the plasma membrane, but no blood vessel is not here because there is another cell. So that's why these are called tissues. So it's not that each cell might have access to the RBCs. So then maybe the oxygen needs to go from the neighbor cell. Okay. So then if you rely on that diffusion, then you need to really reduce the size. Okay. So either the reduce the size or wait for long time. Okay, that's the important. Either smaller distances of size or longer time. Either will work, right? But the longer time is at how long? Is it microseconds, milliseconds, seconds, minutes, hours, days? That's where actually the size of the cell comes into picture. So here actually everyone is seeing this website, right? And send you the link. This is called diffusion time calculator. So this is nice actually program. I like this website. So you can really calculate average time it takes to diffuse a distance of x if you know its diffusion constant. Okay, so if you have a molecule whose diffusion constant is d, and let's say if it needs to diffuse across, let's say, a distance of x, okay, so then how long does it take? It's basically. So this program nicely actually calculates. It's already I have given you the table for proton, sodium ion, potassium ion. These are purely based on diffusion. Okay, and there is one for oxygen, and there is a calculator here. Okay, so we know oxygen diffusion coefficient is 2.1, 10 power minus 5 centimeters square per second. Okay, so imagine, let's say we are talking about a bacterial cell. So roughly the size is 1 micron. I enter 1 micron. And then, okay, let's.
Sir,
yes.
Yes, sir. I was asking that this diffusion phenomena, what you described, is it for multicellular system or for unicellular?
It's nothing to do with cellular systems. It's purely we are assuming that in a medium, okay, whatever medium, diffusion constant is basically specific to that medium. Okay, imagine let's say aqueous medium. Okay, so let's say now just click this button, and it says that okay, now we are asking in seconds. So there's too many decimals, so I'll switch it to milliseconds. Okay, so if I take oxygen molecule whose diffusion coefficient is 2.1 10 power minus 5 centimeters square per second, if it needs to travel a distance of a micron roughly, okay, through by diffusion, how long does it take? It will take about 0.23 milliseconds. Means that if you have a bacterium, okay, so roughly we have oxygen molecule just outside. So roughly to reach the other end is about 0.23 milliseconds. Okay, so this is not too long a time, right? Imagine you have a lot of oxygen molecules, roughly this. But now let's say if you do this in mammalian cells, okay, so roughly what is the size? Let's say the largest we will take 100 micron, right? So then you write 100 microns, and so now it's a large number. Okay, so roughly 100 micron cell. If you have oxygen molecule somewhere here left by an RBC, it will really reach roughly, it will take about 2.3 seconds. Okay, 2.3 seconds.
So
Now we have asked that why can't we have larger cells? Imagine now my cell is 10 times larger than this. Let's say 1000 microns. Okay, so now it will take 238 seconds, and that's really what? How many minutes? 4 minutes. I mean, now you have to wait here at least 2 seconds. Now you have to have 4 minutes. That's really long, because then all the processes inside the cell will be interrupted or delayed dramatically. So all the processes are delayed so much, there's no growth, there's no nutrient uptake or anything. Finally, of course, how the O2 is used. So you have actually a mitochondria. Okay, so mitochondria takes O2 and then will give you. Okay, so adenosine triphosphate ATP. ATP is used for almost any process inside the cell. So you won't have enough ATP generated because you don't have enough oxygen is actually coming. Okay, though we have circulatory system blood, okay, which actually takes nicely oxygen from lungs to every tissue. But the problem is that it will not take inside the cell. It will just leave somewhere outside the cell. But now that oxygen needs to enter the cell. So if the cell is really long, then if it is like 10 micron, then you can really see that how much. 10 micron, it should be much. 10 micron, it will be, okay, 23 milliseconds. That's not really good. It's on an average, you know, so roughly that's much faster than a millimeter. So this is one of the requirements by which basically somehow through evolution we have optimized the cells. Okay, so other way to explain this is that people call surface to volume ratio. But in the end, it's exactly the same thing. It's basically how much, so roughly the size from one end to the other end. Okay, so we are talking about roughly the spherical cell, this one. But you can ask the same question for axons. So axon, now you have to really think about the diameter rather than the length. It's not that oxygen enters from one end of the axon only. No, it through the axon. So cross section of the axon is not really large. So maybe I don't know 10-20 microns. So same as basically let's say diameter of this cell. So this is the primary requirement. Okay. So because of diffusion, okay, you want not only oxygen. So basically, what are the other things which actually you want to rely on? Diffusion within the cell. Within the cell, you want to really diffuse. Diffusion. We are talking about small molecules. It could be large molecules, but if it is really much larger, then you have a mechanism. Okay, so you have what's called active transport. Okay, active transport, where actually what are called molecular motors literally take a vesicle. Okay, so which is present this side of the cell to the other side, or from the center to the periphery, or periphery to the center. Because the diffusion coefficient of anything larger is so small, the time it takes, if it uses only diffusion, too long. It will never really reach the end. So this actually website will give you even the diffusion coefficient of other molecules, you know, like proton, sodium ion, potassium. It's a lovely website actually. You should really play around. Carbon dioxide, urea, glucose, you know. So glucose is a larger size. You can really see that it's almost like 10 times smaller diffusion coefficient. Now you can imagine that through diffusion it might take really long. Okay, but oxygen is actually really important. Of course, your mitochondria is actually spread throughout the cell. Okay, so that also actually solves part of the problem. But then you can't really have then other small molecules which also need to be used diffusion. Then they come. Okay, so try to use this website. Okay, below they have already given for oxygen nicely distance diffusion. If it is 10 nanometers, it takes 23 nanoseconds. If it is 50 nanometer, 595 nanoseconds. If it is a micron, 0.238 milliseconds, like we have seen. 10 micron, 23 milliseconds. Okay. 100 micron, now you're beginning seconds. Imagine if it is 1 millimeter, it's like 4 minutes. Okay, that's too long. So we can't really have anything longer than that. Anything larger than that, then diffusion would not really work. Then you have to have quote unquote blood transport, oxygen transport within the cell, active transport. Okay? But that's not actually how we do it. So that brings basically why the cell size is actually limited. Okay, and this. So yeah.
Should this number matter because it is a steady state?
What do you mean by steady state?
So it means that let's say I want to transfer oxygen from point B to point B.
Okay.
And the thing is that the oxygen which I have to receive at point B, it may take 4 seconds. So it came like 4 seconds away before in point A. But I am receiving constant supply of oxygen at point B because at the point there the oxygen is continuously.
So again, yes. So you have to really see that. Yes, concentration of oxygen is important. Okay, what is the rate at which actually we are taking oxygen through, let's say, RBCs? How much RBC content is there, and what is the capillary volume? All these things are really required. So I think to answer your question, that even if it is steady state supply. Okay, it is steady state supply. I mean, constantly we are taking and then it's releasing and then CO2 is coming back. Okay, but it's only a certain rate at which you can actually pump oxygen rich, let's say RBCs. Okay, so somehow that is also limited. Okay, you can ask that, okay, why not we have, let's say, 10 times rich RBCs? Imagine, let's say, concentration of RBCs is 10 times higher. Obviously, then you have higher rates, right? Yes. But somehow I think so we have really optimized that. Okay, so it's the same thing. It's let's say if outside oxygen is also higher, then of course the loading might be higher. Okay, but all these things I think link to each other. I think somehow optimized at this stage. But let's say if finally it boils down to capillaries. You know how much RBC molecules are there. What is the rate at which blood is flowing? How much it is. Sometimes we see, right, for example, let's say fire accidents. Okay, so there we know that a lot of smoke. Okay, so smoke actually depletes oxygen. So now you're actually breathing other molecules, but there's not enough oxygen. So basically now already quickly everything is, the blood is devoid of oxygen. Then though RBCs are actually flowing through bloodstream, but this is not enriched. Okay, it's the same thing. For example, let's say what you call heart attack. Okay, sometimes people say that after heart attack, if one can revive within certain time, then it's okay, brain is active. But even if you revive after a long time, heart is just a pump. Yes. You know, you can actually in principle restart. But the thing is that brain is already depleted of oxygen. Okay, even after that, even if you pump oxygen, it's not there. Means that that constant supply needs to be there, and somehow I think that really worked down depending on concentration of RBCs and flow rate, outside ambient oxygen percentage, partial pressure. Okay, so this is.
Sir, I have a question that we have in our body we have active transport system for sodium and potassium. So you have sodium and potassium pump. Is it because they have low diffusion constant?
No. So you have to really realize. So O2 can diffuse across membrane.
Yes,
Right?
sir. Yes, sir.
Because
Yes,
it's
sir.
not charged. Now imagine Na+, it cannot because it's charged.
Yes, sir.
And membrane is hydrophobic. It just
Yes, sir.
cannot.
Yes, sir.
So you need an active transport, means that anything which is charged or polar is not going to cross. So basically all the ions, okay, sodium, potassium, are protons, okay? Proton is very small, but still it cannot go through. So you need active transport. Okay, you need to have something bind and then give it through this hydrophobic channel so that it will see this. Okay, there are
Say for any uncharged molecule which has a low diffusion coefficient, so
then,
that should be needed
yes,
active transport.
anything uncharged, non-polar can be embedded into membrane and then they can be taken into the others. Okay, it's a slow process depending on the size of the molecule, but they can be. For example, like most of the time you put fluorescent dyes, you know. But again, if it is non-polar, you have to really keep in mind, if it is non-polar, it will get into the membrane, but it will not be released because it likes membrane, right?
Okay,
right?
sir. Okay, sir. Okay, sir. Okay.
Oxygen, on the other hand, it doesn't really bind to anything. It's non-polar. It goes into the membrane, it passes. It's a miraculous. Okay,
sir.
So this is another website. I think you guys should actually play around. It's very interesting website. It's B10 number 5. Okay, it's a Harvard website. Very interesting information. You can really type in, let's say, largest size cell. Okay, lifespan of RBC. Already some keywords are given like P53 human. Okay, what is the genome size of, let's say, mustard plant? Okay, how many cells are there in human body? You can really ask any of these questions. So most of the time you find actually very interesting information. So you should try it yourself. This is one website. The one at the bottom I already showed you. And so this we have seen largest cell. Okay. So in next 10 minutes, so we want to just quickly go through this. I told you to read the last class. So basically, we will quickly write down what are the components of cell and what are their functions, so that you remember. Okay, this is like practice. You can read yourself, but I won't do it once. Okay. So roughly, you have a cell. I'm writing only. So what you have here, maybe you also should get okay. So it's a cell. Okay. So what do you have inside the cell? So basically you have a nucleus. Okay, what's the function of nucleus? What is the function of the nucleus?
It keeps DNA and protein.
DNA and protein.
It keeps basically the genetic material, keep DNA.
Yeah.
And DNA in the form of chromatin you can call DNA. Okay. And also it might have some proteins. Okay, some proteins which are relevant to chromatin. Typically is chromosome. Okay, but inside the nucleus, there remains a central region called nucleus. Okay, so what's the function of it? Okay, so the central region of nucleus again has a function.
It says ribosomal RNA.
Okay.
RNA synthesis.
So this
Ribosomal RNA synthesis.
Ribosomal RNA. RNA synthesis and basically storage. Okay, And so this nucleus has a
nuclear membrane.
Nuclear membrane or nuclear envelope. Okay. So this is the one main difference with respect to prokaryotes is that is this. So in prokaryotes, the genetic material doesn't surround any envelope or membrane. So it's not separate. So in eukaryotic cells, the DNA chromatin is separated from the cytoplasm. Through this. So that's why it's called true nucleus or eukaryote. In bacteria, you don't have that. Okay. So what's the purpose of nuclear membrane?
Separation.
Okay, separates, let's say, chromatin from cytoplasm. Chromatin from cytoplasm. Okay, so everything outside nucleus is called cytoplasm. Okay, so if you take out all the organelles, then it's called cytosol. Okay, so then what do we have immediately to nuclear pore membrane? So nuclear pore membrane has small pores. Okay, so to allow the material in and out. So occasionally, then it allows some material. What it might allow? So it might allow anything which is copied DNA information into mRNAs. Okay, because of the transcription. These mRNAs now code for proteins. So you need to have something, okay, which needs to be taken outside, right? Something like this. So this is called endoplasmic reticulum. Okay, roughly ER. Okay, but this ER takes, for example, mRNAs from the nucleus and then uses that to synthesize proteins. So to synthesize proteins, you need ribosomes. So these ribosomes are nicely coated onto this endoplasmic reticulum. That's why it looks rough in electron microscopy. That's why it's called sometimes rough endoplasmic reticulum. Okay, so rough endoplasmic reticulum function is protein synthesis. Okay, so most of the protein synthesis occurs on this, and you can see the flow of information very interesting. So it comes from nucleus through nuclear pore complex, and again this is actually bound. You know, all these organelles are independent compartments. And this mRNA actually enters the ribosome. Ribosome synthesizes proteins, and then proteins actually comes out into nicely into cytoplasm. Okay, so anything which is soluble protein, which needs to be in cytoplasm, will be given out by ribosome. Okay, so this so nucleus is actually spherical, and the shape is actually very rigid. You know, lot of proteins. Okay, nuclear lamins. Okay, so this actually gives you nice rigid framework to give the shape to nucleus. Okay, a lot of protein elements. So I don't want to really go into specifics here. Okay, then it will become cell reference. So that gives the shape. Now, so this rough endoplasmic reticulum is actually surrounding the entire nucleus, you know, not just one side, entire nucleus, so that mRNA actually gives this. So if you go slightly below, so rough endoplasmic reticulum, so then you have similar thing, okay? So it's called smooth endoplasmic reticulum, smooth ER. So any information somehow, let's say, needs to be further processed. It's called post-translational modifications. Okay, can be done on smooth ER. So smooth ER, one of the processes which occurs in smooth endoplasmic reticulum is glycosylation. No, actually, smooth endoplasmic reticulum. I'm not sure, maybe not glycosylation. So fatty acids.
Lipid synthesis. Lipid synthesis.
Lipid synthesis. Fatty acids. So lipid synthesis.
Metabolism.
Okay. So why this is separated from there? Because here you need actually protein to direct to synthesize lipid molecules. Okay. So those proteins are actually going to come from your rough endoplasmic reticulum. They have to enter smoothly. Okay. And also you have the other apparatus called Golgi. Okay, again communicating. It's called Golgi apparatus sometimes. Okay, so let's say some proteins which need to be inside the cytoplasm. Okay, fine, they come out of your rough endoplasmic reticulum stuff. But some proteins need to be embedded in your membrane. Means that they are hydrophobic in nature, right? Okay, so now these molecules, if you leave just in aqueous environment there, they will never really reach this. They'll come together, aggregate, and then they won't be happy. So you need to really direct them. That's a very tricky part, because from the synthesis onwards, the moment the molecule is made, it needs to be in hydrophobic environment. Okay, so all as the proteins are actually being synthesized, okay, they are directly going into so this again compartment. Okay, there's a transfer, and then finally they'll come up with small vesicles. Okay, so in these vesicles, the protein is actually embedded, and then this vesicle finally combines with this membrane. Okay, so vesicles are nothing but again small lipid bound spheres. Okay, where inside is aqueous, but it's bound by lipid and these proteins are actually embedded on this lipid membrane. This vesicle actually now fuses with your plasma membrane. Okay, so once they are embedded, then they are stable. Okay, see how the flow of information from the nucleus, protein synthesis. Sometimes proteins can come out directly, but if proteins are nearly embedded in membranes, then automatically small vesicles bud out and then go into Golgi. And then Golgi knows that where they need to be exported. Sometimes they need to be embedded, okay, but sometimes they need to be exported. Okay, so then there are signals. Okay, so these peptides, like a stamp on a postcard or an address on a postcard, okay, is that this envelope needs to go this address. So little proteins have small peptide sequences called signal peptides. These peptides tell the Golgi that they need to be packed and then export whichever address.
Sir, what did you say about the vesicles? What are they filled with? Hello?
Yeah, just a minute.
Yeah.
Sorting and then center. Oh, sorry, yeah. Who is it? Pratik?
Yeah, it's me, sir.
Pratish.
So, what did you say the vesicles are made of? What is the composition?
Vesicles are made of basically lipid molecules, okay, in a bilayer fashion. So interior is aqueous and outside is aqueous, but there is a boundary. The boundary is made up of lipid bilayer. So this lipid bilayer same as actually plasma membrane, but however these lipids will have very selective proteins which are targeted for membrane. Okay. Plasma membrane will have a lot of other proteins and then cholesterol and a lot of other. Okay. Okay, so that's this. What else we have? Okay, so let's say if proteins are not good, okay, our proteins work is done. Okay, so you need to get rid of these molecules which you don't need them anymore. Okay, so how do you get rid of them? So then you have okay, so lysosomes, anything which is unwanted. Okay, degradation. Degradation of proteins, any other molecules which need to be recycled. Okay, recycled inside lysosomes. Again, this is membrane biology. Okay, so again, there is a huge machinery. You know, you don't want to really recycle the right molecules. Somehow they have signals. You know, so these signals will tell them there's something called autophagy. Okay, so these signals will tell which molecules need to go. Okay. How all these processes are actually running? We need energy, right? So we need ATP. ATP is the currency for any of these processes, from protein synthesis, DNA replication, ribosomal RNA synthesis, anything. You need ATP. So cell needs to be filled with ATP. So you have, okay, mitochondria floating around. Throughout the cell. Okay, so 8. Okay, so uses oxygen and then generates ATP. Okay, so you have all the machinery which is required to convert. Okay, your O2. Okay, and using glucose to ATP is occurs in mitochondria. Mitochondria spread throughout the cell. Okay, and you have seen that axon in neuron cells. Entire axon is filled with mitochondria as well, because you need energy throughout. Because you need ATP throughout the cell. Okay, what else are we missing?
Ribosomes.
Ribosomes, yes. Okay, so we mentioned ribosomes here. Okay. So, for example, they are mostly around endoplasmic reticulum, but they could be free ribosomes and stuff. Okay. So ribosomes are protein synthesis factories. Okay, protein synthesis. Okay, and now the shape of the cells. Okay, so like RBCs have one shape, and neurons have another shape. Other cells might have other shape. Muscle cells will have elongated shape, right? So how do you provide the shape, okay, to the cells? So then you have a lot of cytoskeletal elements. Okay. So these filamentous proteins, okay, so they give actually the framework or the shape, okay, or the skeleton to cell. Cytoskeleton. Okay, so this actually has 3 different types of molecules. One active filaments,
microtubule.
Okay. actin filaments, then microtubule filaments, and then intermediate filaments. Okay, so they have actually 3 different types of filaments. Their jobs are different. Actin filaments uses motor proteins, kinesin. Microtubule uses dynein. Okay, so these are motor proteins. To take cargo from one place to the other place inside the cell. So this is the active transport, which I was talking about. Okay, so I told that these small vesicles, which are taken from bullseye to, let's say, periphery. Okay, if you rely on diffusion, it will never really work. So you need these motor proteins. Okay, so which are connected, let's say, from center to periphery. So then these vesicles bind to this motor protein, kinesin or dynein, and then they can actually walk course. And these are directional. If I remember correctly, kinesin takes stuff from periphery to the center of the cell. Dynein exactly does opposite. So it always takes the cargo from the nucleus to the periphery. They work exactly opposite. Intermediate filaments will give you basically again rigidity, which don't really use any of the motor proteins. So that will give you the stiffness to the cell. Okay, right stiffness. So that cell has certain shape, so that it doesn't become, if it is too fluid, then basically we don't have the rigidity of the cells. Okay, so that's things. Let me just check if I missed anything. And finally, of course, plasma membrane, which is again important. So plasma membrane has a lot of proteins, okay, which can take material from outside to take it inside, or transport material from inside to outside, including ion transport, or proton transport, sodium transport, potassium transport, calcium transport, or glucose transport. Okay, so because glucose, it's not through diffusion, you have it. So anything which needs to be. In addition to that, there are a lot of other proteins which actually act as a signal. Okay, I think roughly, I think that's it. So if I missed anything, I think more or less. Okay. Of course, plant cells will have slightly different, but you don't have to worry too much. You can actually do that. Okay.
Peroxisomes. Yeah, peroxisomes.
Okay. So. Okay, what do peroxisomes do?
Oxidation of fatty acid.
Oxidation of fatty acids.
Oxidation of
Fatty acids.
okay. Okay, I think that's last. Okay, so oxidation. So, roughly, you need to see that. So in eukaryotic cell, because you don't have all these things, okay, not as enriched. So the things process become much simpler. Though of course the central dogma transferring information from DNA to RNA to protein is there, function of the cell is there. Lot of taking energy from outside, food from outside is there. But you don't have such specialized things because first of all the cell is very small, okay, and you don't have any nuclear compartment, so everything is free. So half the volume of the cell, okay, so it's basically DNA and ribosomes in bacterial cells. Most of the volume is occupied by ribosomes because you constantly synthesize proteins, okay, and genome is very small. Okay, they have only about 4,000 proteins, okay, but we have much richer protein repertoire, okay. So that actually sums up roughly this class, the components of cell, okay. So this picture.
Sir, yeah, sir, what you are talking about, the sodium and potassium ion active transport. So are the proteins of this cytoskeleton involved in all the kind of active transport there?
No. So inside the cell, the cytoskeleton elements uses these motor proteins, kinesin and dynein, to transport variety of cargo. Okay. So most of the time, this cargo is embedded in a vesicle. The vesicle inside can be anything. It's just like sending some cargo to basically a cargo process where you don't know. Everything is filled in a box. Okay. The postman doesn't know what is inside, but postman knows that okay, the address is here. It needs to be taken. So these motors, okay, know exactly which way they are actually going. These are literally like intracellular rail tracks. Okay, so these motors will have only cargo hold. The cargo fits in, cargo goes. But inside the cargo could be anything. Most of the time, these vesicles, either you move the proteins from ER to membrane, proteins from, let's say, unwanted proteins to lysosome, some other thing to, let's say, some other things. Okay. So again, concentration of the nutrients, concentration of the molecules inside the cell is not uniform. Okay. So actively somehow you change the concentration depending on cell type and cell state, etc. But the sodium and potassium, okay. So these are just only import machines says that okay, potassium is just allowed inside, but once it is inside, there is no machine to say that okay, you take the photon. It's purely diffusion. Okay, but these molecules are actually through the cell, right? Through the cell membrane. So it's actually the input comes from all sides. Okay, and then you balance sodium and potassium. Okay, these are voltage gated ion channels. So they know exactly the potential difference between inside and outside. They regulate. Okay. So the sodium and potassium is very tightly regulated. If I remember correctly, sodium inside is very high, potassium outside is very high. Okay. So you need to actually maintain that. Okay, tight regulation. So these ion transporters, okay, so tightly regulate that. They know that if there is a gradient of potassium, then only they open. Okay, and there is something for proton. And imagine there is something for water. Okay, there are something called aquaporine. Aquaporine is a molecule which, okay, transports water molecules, okay, inside to outside, outside to inside. Imagine, let's say, because you have the salt gradient is there, you know, proton gradient is there. So many things you need to really maintain. Okay, for everything you have a regulator. Besides, you need to maintain the total volume of the cell. You know, you can't really increase the volume of the cell, then things will get diluted. If you don't have enough membrane, it will burst. Okay, if it is like if you take out water from inside the cell, then it will actually spacious, then nothing can be living. You know, so you have to tightly regulate all these small molecules of sugar. Okay, so and you have for everything you have a machine in the membrane. Okay, just only for O2 it relies on diffusion. Okay, or any other small organic molecules. Yes, depending on nitric oxide also, I presume maybe diffusers. I'm not sure. Nitric oxide is basically a gaseous. What is it? Messenger molecule. Okay, so there are very few gaseous molecules. Okay, which are important. Ammonia, for example. Okay, so it's important. O2, of course, well known. NO, nitric oxide. Okay, so aquaporin, there is a Nobel Prize involved. So the guy who discovered how exactly water is transported through protein molecules. Okay, so this class now summarizes components of a cell, their functions. So next class onwards, so far no chemistry, right? It's basically still bird's eye view. Okay, so next class onwards we go into the chemical entities. Okay, so which molecules actually make what? Okay, how these molecules important and what are their important roles. So just one slide. Because these pictures, for example, like previous pictures, everything has been very nicely in this quarter. But this just cytoskeletal elements is one thing which you should appreciate is that the one on the left, you see, the blue spheres are actually nucleus, and you can see the periphery, how random is the cell shape, and what you see in green is microtubules, which use dynein, okay, and the one in yellow, no, the one, okay, left side figure, the one in red, again, I think the one in red is actin, I think, okay, yeah, okay, the one in the red is, yeah, actin, these are stress fibers which use kinesin, okay, and the one on the right side, you can clearly see that the red mesh is intermediate filaments, the third one, which is actually made up of a protein called keratin. And also you can actually see nicely cell division, color coded. Okay, so the blue are basically the chromosomes are being actually separated. So this is actually arrested cell division. Okay, you have actually two kinetochores, okay, separated. Okay, so those two dots are actually this. And the green lines are actually microtubules. So one of the ways is that microtubules are used to transport the content inside the cell, and also they come handy when you actually cells are dividing. You have to literally mechanically separate cells. Okay, at the same time you have to divide perfectly. Okay, we have, let's say, chromosomes, right? Every pair needs to be sorted out. If you don't get sorted out properly, then your cells will not function. One cell will have more chromosomes, another cell will have less. Then both cells are actually equally bad. Okay, so the blue are actually nice chromosomes, and then green is green is microtubules. The red is actually intermediate. Okay, just I wanted to show you so that you appreciate how complex is when we say the cytoskeletal elements are inside the cell. Literally the entire cell outside the nucleus. Okay, I think I covered whatever I wanted to do in this class. So next class we will start small molecules and macromolecules. Okay.
Sir, you mentioned the actin filament and microtubules works with the kinesin protein and dynein protein respectively. Can you please again mention what are the directions of their works?
Yes. What is the last part? Could you repeat the last part?
The directionality of the work of kinesin and dynein protein. I want to know that. You mentioned that, but I forgot that.
Yeah, they're directional. Okay. So if I remember correctly, they are unidirectional because these filaments have what are called positive and negative end. Basically, they are asymmetric. Okay, they are not symmetric. So kinesin, if I remember correctly, uses actin filament. Okay. So kinesin walks in this direction. Means that cell membrane to nucleus because the filaments are oriented. The microtubule, on the other hand, goes from nucleus to the periphery because this track is different from this track. Okay.
Okay, sir.
I know, when I was a kid, we used to have for the railroad different gauges. So one is narrow gauge, another is called wide gauge, medium gauge, something like that. Okay. So you have a rail which has that spacing, correct? So exactly same thing for acting. The track is right for acting, so dining doesn't fit on acting, and kinesin doesn't fit on nitric keyboard.
Okay, sir. Okay.
But interestingly, they actually crossed up, though they are actually going on different tracks. They are crossing. Okay? Very interestingly, they actually caught. Okay. That's yeah. Okay.
Okay.
So I have a question.
Yes, Tushar.
So these sodium and potassium ions, they are prone to get hydrated. So is that the hydrated sphere of that ion goes inside the cell, or that the naked ion suspended which goes inside the cell?
Yes. So people have actually sorted out. So we have crystal structures of sodium channel and crystal structure of proton channel, crystal structure of potassium. Okay, so almost all these channels recognize what do you call it? Waterborne, let's say ions. Okay, aqueous ions. Okay, at the edge, you can really call the mouth of the channel, the pipe. But then, as the ions go inside, they strip these water molecules. Okay, they detect aqueous ions, but they don't really take the ion along with water molecules to the other side. They have channels, so they bound aquated one, but they strip water, but they allow only ion. Okay, as it goes through intermediate states to reach the other side, it actually mimics the water environment. Okay.
Yeah.
And directionality is actually equally important. It's not that they go both ways. No, these are voltage gated. So they open actually only. The gate opens only in one direction. So they know which direction. I think it's true for all. Because otherwise, imagine if proton, which one is smaller? Sodium, potassium, proton. Put it in the order, small to large. Which one is small?
Proton.
Proton.
Basically, hydrated sphere of potassium will be smaller.
Potassium will be small.
Potassium
hydrated potassium will be smaller.
Imagine all are hydrated, okay, or non-hydrated. What is the size?
Then sodium will be small.
Non-hydrated for
Non-hydrated
proton will be
sodium.
small. Proton will be small, then sodium, then potassium. For hydrated,
Then sodium, then potassium.
Potassium will be the smallest, and sodium will be the.
Okay. So now you take whichever is largest. Let's say imagine hydrated potassium is large. So you have a larger channel, right? It needs to fit. Now exactly proton is supposed to go because it's a small volume, right?
Yeah.
Right? But it will not go through. The moment it will go this protonated one, it knows that it's actually, it's like a track, So somehow the ion needs to be detected by 4 sides, okay, equidistant. So now if it is detected by only one side, okay, a small one cannot really fit here, right? Imagine like crown needles, okay? So they don't fit on 4 sides. If it fits only one corner, that doesn't allow. So it's actually the mechanism only works only if all 4 sides fixed, then it pushes. If
Sir, we are not able to see your fingers. I mean, yeah.
Okay, so imagine, let's say I have 4 tips, 4 fingers. Okay, it's a perfect square. The ion fits in the center, interacts with the 4 fingertips, then only it will be pushed forward. Okay, but if the ion is, imagine let's say now you have a larger channel, potassium channel, but somehow protonated proton, okay, H3O plus, will bind only one corner because it's not really large enough to see all 4 points. Now this pushing mechanism doesn't really work because it's stuck to only one side. It can be pushed efficiently if all 4 do this. Okay, so that's actually how the water will not really go to your larger channels. Okay, so every channel has crystal structures. Okay, we have beautiful crystal structures in bound, and actually the ions are being pushed in the channels. Okay, so through crystal structures we know a lot of course. Okay, good. I think that's a good start of biochemistry, biophysics. So next class onwards, I think we will go into small molecules and large. Okay. So yeah, so I'll put these slides as well as lecture 2 and lecture as well. Aditya, you are still there?
Yes, sir.
Okay. So yeah, I think that's all. And maybe I'll make one assignment based on this. But these website links are there in the PPT. You just try this. The diffusion calculator is very interesting. This Harvard website, Bio Numbers, beautiful. A lot of information. You know, you can really ask questions like how many hairs are there. You know, what is the longest living species? How many species actually are there? You know, what is the longest living spider? You know, you type in, more or less you get very interesting information. It's basically numbers. And the one at the bottom is an article only. Okay, just to give you insight that how actually people are thinking. You know, so cells replacing from one living thing to another living thing, they are still living. You know, they are living longer than the primary the donor. And basically, this is just to give you interesting thought that. It's very interesting that, you know, in egg, the yolk is actually suspended exactly in the middle. It doesn't really float up and down. So because something is called chalazan, C-H-A-L-A-Z-E, that actually anchors the yolk in the middle. Amazing. Okay. Yeah, I think we need to add your numbers to WhatsApp. We have created a group, but I thought Jerry would forward, but looks like he's not able to do. Can one of you guys forward your numbers to Aditya so that he will add who are actually taking this course? Then, for example, these kind of sites, when I find, you can just put it in WhatsApp so that if you can click and then you can actually do some interesting stuff. Okay, that's all. Okay, thank you and bye bye. See you on Monday.
Thank you, sir. Bye.
Thank you, sir.
Thank you, sir.
Bye, sir.
Monday is next class. Okay.
Okay.